This video is going to be about the primary, secondary, tertiary and quaternary structures of proteins. There are 20 different amino acids which are used within the cells of organisms. Amino acids are made up of two groups. The first is a basic amino group. The second is an acidic carboxyl group. These two groups allow single amino acids to bond together into long chains called polypeptide chains. The bond which occurs in polypeptide chains is called the peptide bond. This bond forms between the NH2 and the COOH of a second amino acid. An amino acid also contains an R group. This is what differentiates all the amino acids. Depending on the molecule which replaces it, determines which amino acid will be there. It will be amine. Peptide bonds are formed from a condensation reaction. This reaction, as it forms a peptide bond, a peptide chain, forms H2O molecule. Hence the name condensation. This reaction is a result of an enzyme catalyst. The breakdown of these bonds is also created by enzyme catalyst. This reaction is called hydrolysis. The unique pattern which the amino acids make to form a polypeptide chain is called the primary structure. Chains which are made up of 50 or more amino acids are called peptide chains, while chains which contain less than 50 amino acids are referred to as peptides. A protein is made up of one or more of these peptide chains. After the peptide chains have been synthesized, they form different shapes, which are folded or pleated. This folding shape is called the secondary structures. There are two very common secondary structures of proteins. These are known as the alpha helix and the beta pleated sheets. An alpha helix is held in a spiraled shape by hydrogen bonds between the CO of each peptide bond and the hydrogen of the HN within the peptide bond for amino acids below it within the helix. A beta pleated sheet consists of paired strands lying beside each other. The hydrogen bond in the beta sheet occurs between the carboxyl oxygens of one strand and the amino hydrogens of the strand it is paired with. These paired strands can either be parallel or antiparallel. This is dependent on whether the strand's direction is the same or the opposite of each other. The beta sheets that are antiparallel are more stable as the hydrogen bonds formed between the two strands are more aligned. The strands within the alpha helix and the beta pleated sheets are the primary structures of proteins, the polypeptide chain spoken about earlier. The tertiary structure of a protein is what describes the folding of the chains into a 3D shape. This shape includes the primary and the secondary structures, alpha helixes and beta pleated sheets. The 3D shape of the tertiary structure is dependent on the sequence of amino acids of the primary structures. Which produce the secondary structures, alpha helix and beta pleated sheets. The 3D shape is also dependent on whether the amino acids within the chains are hydrophobic or hydrophilic. If a chain contains amino acids with a hydrophobic nature, then they will tend to point inwards especially with globular proteins, as they are normally in an aqueous solution, or surrounded by water. Tertiary structure is maintained through the use of disulfide bridges. The amino acid chains fold and are held together via attractive forces. The disulfide bridges are the covalent bonding of two sulfur containing these residues. These bridges tend to occur more frequently in the structural proteins, as they will contribute to the overall strength of the structure. Amino acids which are further away from each other within the polypeptide chain or in different secondary structures within the tertiary structure can interact with each other, and the different amino acid sequence in the primary structure will affect the folding of the tertiary structure. The shape of the tertiary structure changes depending on the sequence of amino acids which create the primary structure, because the polypeptide chains will create the most stable form they can through the use of bonds such as hydrogen and covalent bonds. The different amino acids within a sequence will change how many bonds there will be within the chain, and this will therefore change the structure of the tertiary structure. The tertiary structure is when the different R groups which differentiate the different amino acids interact. The properties of the amino acid and the polypeptide change depending on the different R groups of the amino acids within the polypeptide. For example, hydrophobic amino acids such as glycine and alanine will pull inwards within the structure, while hydrophilic amino acids such as asparagine and glutamine will be drawn outwards. Amino acids such as cysteine will form a covalent bond between two sulfur molecules, creating disulfide bridges spoken about earlier. The final structure of a protein can be the tertiary structure. But when it is made up of one or more tertiary structures, the last molecular structure is known as the quaternary structure. The quaternary structure is the fourth level of structural organization. These structures, as well as containing the primary, secondary, and tertiary structures, can also contain non-protein prosthetic structures. The shapes formed from the quaternary structures are important for the different proteins to be able to complete their functions. There are two different types of proteins which these structures will create. These are either globular proteins or fibrous proteins. Fibrous proteins are used for structure and support, while globular proteins are used in transport, for example hemoglobin carrying oxygen, and for communication within the body, for example, hormones. The change of the primary structures of proteins, even the change of one amino acid, can have an extreme effect on the structure and function of the overall protein. An example of this is hemoglobin. If one of the glutamic acid residues are replaced with a valine in each beta chain, this will cause a condition known as sickle cell anemia. This causes the hemoglobin in the red blood cells to be less effective in absorbing oxygen and can cause blood vessels to be blocked, which can be fatal. What is insulin? Insulin is a hormone produced and stored by the pancreas. Insulin is a globular protein. Insulin has several functions within the body. Its main functions include maintaining and regulating the use and storage of glucose and allowing cells to get the glucose from the blood for energy. Insulin's primary structure consists of two polypeptide chains. This means insulin is a dipeptide. Insulin is made up of 51 amino acids, 21 in the A chain and 30 in the B chain. These two chains form secondary structures through the use of hydrogen bonds. The A chain of the insulin forms two secondary structures. Two short alpha helixes which lie alongside one another, and the B chain which contains one secondary structure, a long alpha helix. These chains are held together through the use of disulfide bridges. Two disulfide bridges connect the B and A chains together, and a third disulfide chain bridge is formed between the glycine residues on the same chain. The A chain. The secondary structures, when put together, create the tertiary structure of insulin. When one or more of these dipeptides, tertiary structures, are put together, they form a quaternary protein, such as a hexamer, which is three pairs of tertiary structures of insulin put together. The interior structure of an insulin molecule contains non-polar side chains, while the exterior structure consists of polar side chains. This allows the insulin to be soluble in water, as the polar side chains can create hydrogen bonds with the hydrogen atoms within the H2O molecules. This structure relates to the function of insulin, as it allows it to be transported around the body within the plasma of the blood. A second function of the structure is that insulin molecules can form granules made up of a hexamer. This granule includes three paired insulin tertiary structures surrounding two zinc ions. This structure is related to the function of insulin, as for insulin to work, it needs to be able to interact with the receptors on the cell membrane. The insulin receptors are made up of two alpha subunits and two beta subunits. The alpha subunits are the active sites of insulin. The alpha subunits are outside the cell membrane, while the beta subunits penetrate through the membrane into the cytoplasm. Disulfide bridges connect the alpha subunits with the beta subunits. There is also a disulfide bridge which connects the two alpha subunits together. A hexamer structure of insulin is required to activate the insulin receptors of the cell membrane, as shown here in the diagram.